Carbohydrate in Food, Part 2. The course outline for this part starts with the sources and structures of oligosaccharide and polysaccharide, their physicochemical and functional properties, and starch gelatinization and retrogradation. Oligo and polysaccharides, both of them are considered as complex carbohydrates because of their large molecular weight. Oligosaccharide consists of 3 to 9 monomers. All of oligosaccharides are basically indigestible. What it means by indigestible here is our human's digestive system don't have the enzymes that can actually cleave the monomers to break it apart. That is why all these saccharides are indigestible except for only one type, so-called maltodextrose. The benefit of the indigestible carbohydrate is it can promote the growth of the intestinal bacteria in our large intestine, and then these bacteria will break down these fibers into a smaller or less complex carbohydrate. This will protect the lining of our intestine and can prevent cancer. So basically all these saccharides are prebiotics. This table shows different types of oligosaccharide with their respective sources. Legumes are the main source of oligosaccharides such as beans, lentils, chickpea, soybean. Fructooligosaccharides and inulin highly contained in artichokes, chicory, leeks, and onions. In Malaysia we eat a lot of onions, so it's good for us. Polysaccharides exist as 2 types, homopolymers and heteropolymers. Homopolymers consists of only one type of monomer. For example, cellulose and amylose that consist of only D-glucose, but they differ in terms of the linkages of the glycosidic bonds. For cellulose, the glycosidic bond is actually beta-1-4, while for amylose, it is alpha-1,4 glycotic bond. Hope you still remember the positioning of the hydroxyl group case in our previous lecture. Meanwhile, heteropolymers have more than one kind of monomer exist in the polysaccharide. For example, carrageenan, which is a copolymer where it is alternated between alpha-1,3 and beta-1,4 beta-D-galactopyranosyl linkage. For block polymers such as alginate, where it's just like one whole block is just beta-D-manuronic acid followed by the whole block of alpha-L-guluronic acid. Next is branched copolymers such as guar gum. The backbone of the polysaccharide only one type of monomers, and then they are branched with another kind of monomers. Final type is random copolymer, a mix of different kind of monomers inside the polysaccharides. Polysaccharides are used in food industry in the form of food ingredient to modify the physical and chemical properties of the food depending on what type of targeted properties that the food industry desire. They can form gel, stabilize emulsion, and control the water activity. Polysaccharides also exist in several conformations. Some are helices with different pitches and numbers of monomers. Some are extended relatively flat ribbons like in cellulose. Some have irregular flexible coils when there are alpha-1,6 linkages and double helices. So, it depends on the different type of polysaccharides. For example, pectin is more branched than glycogen, which has much smaller monomer units right per branch. These are several types of polysaccharides that we have, and we are going to look into one by one of their sources. The most popular polysaccharides is the plant reserve polysaccharide, which is starch. Cereal starch can be found in rice, corn, and wheat. Tuber starch in potato, tapioca, sweet potato, and yam. Pith starch and sago, while legume starch can be found in legumes such as mung bean, chickpea, pigeon pea, and cowpea. Then we have plant structural polysaccharides such as cellulose, hemicellulose, and pectin, which can be mainly found in the cell wall of the plant. Next, plant exudates are gums such as gum arabic from acacia plant. Other than plant exudate, there are also gums from the seed of the plant such as guar gum, locust bean gum, and tara gum. There are also polysaccharides from algae such as agar-agar and carrageenan from different species of red seaweeds, and alginate from brown algae. Last but not least are polysaccharides, which are being produced by microorganisms such as xanthan gum that is produced by fermentation using the bacterium Xanthomonas campestris, and gellan gum produced by fermentation using the bacterium Pseudomonas elodea. Starch is the most famous polysaccharides used in the food industry as human food as food ingredient. They are originated from plants as a reserved energy, and it is used as our source of calorie for humans. They exist in endosperm and tubers and roots and fruit and stem and cereals. Some of them are digestible and some are non-digestible depending on how they are being digested in our digestive system in our stomach and intestines. Then it will also influence the texture of food. This table shows that different starch found in different sources of foods have different in size of granule and shape or organization of starch granule. Meanwhile, these figures show the shape and organization of starch granules from different types of the starch under the microscope. The composition of starch. The monomer for starch is glucose, right, and it actually exists as in a linear chain of alpha-1,4 linkage, which is the amylose, and also branched by alpha-1,6 linkage, which is the amylopectin. Amylose is the minor polymer in starch. It exists around 20 to 30% by nature. Amylose has molecular weight around 300,000 to 1 million OK. It is linked together by the glycosidic bonds at alpha-1,4 4 linkage and amylose can form a very strong interaction between themselves and other kind of carbohydrate polymers, and they form very strong gel and films. Properties of amylose. It has a helix formation, which is a very stable conformation. It is a left-handed, means the helix to the left, and then every repetition of the turn has 6 anhydrous glucose unit. Amylose has hydrophilic region to the outside and hydrophobic region to the core, which is inside the helix core. Molecules can be trapped inside the helix core, such as iodine, organic acid, alcohol, and lipid to form amylose complex. This figure shows the structure of starch, where we have the straight branch of amylose that are connected together by alpha-A1, 4 linkages, and then the branches of amylopectin will be connected by alpha-1, 6 linkage. Why it is alpha-1, 6? Remember the numbering of the carbon in the previous lecture. Here, the carbon number 1 of glucose from amylopectin and carbon 6 of glucose from amylose connected together. Properties of amylopectin and starch. It has a very large structure, as the molecular weight can be up to 100 million. It is highly branched, and around 5% of all the bonds are alpha-1, 6 branch points. It has 3 levels of branching. The C chain has multiple branch that are the initial branching point of amylopectin, and then B chain carries 1 or more branch points, and the one without any branches is the A chains. They are not branched randomly. They are very organized in a way, and it is called a bottle brush model. It is a bottle brush model, as they have done dense branching in clusters with 1 reducing ends only, and then all branches on the opposite end does not have any reducing ends. This determines the interaction of amylopectin with itself and other types of amylose. Moving on to starch gelatinization and retrogradation. Starch gelatinization is the collapse of molecular order within the starch granule manifested in irreversible changes. It will cause granular swelling, native crystalline melting, loss of birefringence, starch solubilization, and viscosity development, which will be explained in the next slide. So what happens during just the gelatinization? In A, starch granules prior to heating in excess water means no gelatinization happens. Figure B is the starch granules after heating to a temperature in excess water that melts the native crystalline. Figure C shows the starch granules swell more during the processing because they absorbed water and therefore more gelatinized. If you can focus that there is a loss of birefringence, which is the cross shape or Christian symbol of the starch granule that are caused by the organization of the starch in figure A and B, but no more in figure C and figure D. Highly gelatinized starch granules are ruptured due to overprocessing. The starch granules completely disappeared. There is leaking of amylose and amylopectin, but mostly amylose, and changes in the flowability of the starch. Before gelatinization, it doesn't dissolve in water, but then with gelatinization, as the temperature increase, it will start to dissolve in water, and then it will start to increase the viscosity again. This graph shows changes in starch during and after cooking. During the initial graph, the temperature increases up to 95 degrees Celsius, and then hold it for a couple of minutes, and then the temperature will reduce again around 50 degrees Celsius, or almost back to room temperature. When the applied temperature exceeds the starch granule gelatinization temperature, swelling and partial rupture of the granules is initiated, shown by an increase in viscosity. Thus, the pasting temperature and the peak viscosity represent the most meaningful values for an unspecific description of starch gelatinization, whereas Tp represents the beginning of gelatinization and Pv the intensity of gelatinization, both viscosity-based. As the temperature goes down depending on what type of starch, there is possible formation of reassociation of these molecules to organize again, and this is what we call as retrogradation. Depending on types of starches, reassociation of molecules to form gel requires starch with high amount of amylose. Meanwhile, the non-retrogradation is much more stable starch with higher amount of amylopectin. This is the summary of how gelatinization happens. When gelatinization temperature is reached, water will enter the granules. The granules will swell and viscosity of the starch paste will increase until peak viscosity is reached. The granules will then crack or rupture, amylose will leach out, and viscosity of the starch paste will decrease. When the paste is cooled, the amylose will start to align close to each other and form hydrogen bonds. Viscosity of the paste will start to increase again, and this phase is known as setback. If the starch concentration is high enough, starch gel will form. Factors affecting starch gelatinization: starch sources, because different kind of storage have different amount of amylose and amylopectin. The water content: with higher amount of water you will have higher gelatinization rate. Heating rate: the faster gelatinization happens. Lastly is the presence of other components such as salt, sugar, lipid, protein, and polysaccharide. Taking sugar as the example, sugar will increase gelatinization temperature because it competes with starch for the available water, and also it will inhibit the starch swelling because the sugar and the starch will fight with each other to get water. Once the sugar is attached to the hydrogen bond of water, it will cause the sucrose molecules bridging between water by hydrogen bonding, so this will limit the movements of the starch granules by the sugar-water hydrogen bonding. Let's talk about pasting properties. Pasting is the phenomenon following gelatinization in the dissolution of starch. It involves granular swelling, exudation of molecular components from the granule, and eventually, total disruption of the granules. Starch with high amylose content form gels more easily than do starch with high in amylopectin. Paste derives from high amylopectin starches considered to be non-gelling but have a gummy, cohesive texture. It is useful as thickening agent due to its ability to increase the viscosity of solution. Next is retrogradation. Starch retrogradation is the event that occurs when starch molecules begin to reassociate in an ordered structure. In its initial phases, two or more molecules may form from a simple juncture point, which then may develop into more extensively ordered regions. Ultimately, under favorable conditions, a crystalline order appears and precipitation from solution occurs. Focus on the red keywords here. So, it is a process when the polymers comprising gelatinized starch begins to reassociate in an ordered structure. The linear amylose molecules are attracted to each other and form bundles of parallel polysaccharide chains by the formation of hydrogen bonds between hydroxyl groups on neighboring molecules. Retrogradation is evident when amylose-containing starches are cooled, resulting in water release. This figure illustrates gelatinization and retrogradation phenomenon happens on bread from dough until steel bread. At dough stage, it has amorphous amylose, polar lipid, and crystalline amylopectin in one starch granule, hold together with other granules by gluten. Once the dough is being cooked, you can see that the granules start to swell into larger size or loosen up, where they lose their organization and their ordered structure. There is formation of retrograded amylose and gelled amylopectin that are leaching out from the starch granules. Amylose complex is formed between amylose helix and polar lipid that contributes to the sensory properties of fresh bread that has the crunchiness on the outside part and soft inside. After some time, or we called the process as aging. Let's say if you have the fresh bread and you just leave it outright, whether it's in container or in open air, the bread will become harder, the texture will become much more chewy. This is because the retrogradation process happens. In stale bread, the fresh bread, the ordnance structure is like completely gone, but then they start to really organize and reassociate again inside the starch granules as well as between the granules. So let's say you have a stale bread and you put in a microwave for a couple of seconds. It will start to disrupt again and you will have a softer bread, but if you leave it again, it will have retrogradation again and will form a hard bread again. In this final slide is to highlight the difference between gelatinization and retrogradation. Retrogradation cannot happen without gelatinization. So gelatinization happens by heating the starch in excess water that cause disruption of ordered structure, and then with this ordered structure plus time and can develop an ordered structure again, which is retrogradation. Coming to the end of the lecture, the students are expected to differentiate between oligosaccharides and polysaccharides based on the structure and functional property, as well as the important chemical changes happens on starch during starch gelatinization versus starch retrogradation. These are the references. Thank you.